So welcome to our series, Translational Research in Bacteriophage Therapy. This is a seminar series that is sponsored by the Center for Innovative Phage Applications and Therapeutics, the IPATH at UCSD, as well as University of California, Irvine. Our goal is to bring together researchers who are interested in bacteriophages, as well as those who are interested specifically in phage therapies, in the hopes that our conversations can ultimately result in improved bacteriophage therapies. Our speaker today from the Baylor College of Medicine is Dr. Austen Terwilliger. Dr. Terwilliger is originally from a small city in Pennsylvania. He got his bachelor's degree in biology from the University of Pittsburgh. And since then, he's moved down to Texas, where he's been working with Dr. Anthony Moresso at the Baylor College of Medicine. His original doctoral work was looking specifically at Bacillus anthracis, where he was investigating genes involved in hemoglobin amino acid acquisition. He continued, after getting his PhD, he continued as a postdoctoral scholar with Dr. Moresso there, but in his postdoctoral work, then moved into work specifically on bacteriophage therapies. And as a postdoc, he did a lot of work really looking at identifying bacteriophages that target escape pathogens very specifically, which is something that we're very interested in here at IPATH. And they've been working on things like developing phage cocktails that target the escape pathogens that can be used in phage therapies for compassionate uses. Dr. Terwilliger is also responsible for inventing the Modify device, which maybe some of you guys know about, that sort of helps them direct phage evolution against very specific targeted bacteria pathogens. Dr. Terwilliger has taken a slightly different path than maybe some of our other speakers. He has stayed at Baylor for really quite some time now, and he helped to found the Taylor Lab at Baylor. And let me read this, because Taylor stands for Tailored Antibacterials and Innovative Laboratories for Phage Research down at Baylor. The one thing that I really want to point out is that Dr. Terwilliger's work really at Taylor has been so instrumental in helping the things that we do at IPATH. And quite frankly, Dr. Terwilliger, even though he probably wouldn't say it, is probably one of the world's foremost experts in terms of purifying bacteriophages for use in bacteriophage therapies. So we're hoping that we can learn quite a bit from some of the work that he has been doing in purifying bacteriophages, and hoping that perhaps we can even adopt some of those things in the future. Of note is that his Taylor Lab at Baylor is very interested and is in the process of developing a GMP facility. So not only are they producing basically purified phages today for folks like us that are engaging in bacteriophage therapy, but they're positioning themselves potentially to be a centerpiece of bacteriophage therapies for probably many years to come. The other thing that I would mention is, while we're very interested in the Taylor Lab as a place where you can basically get bacteriophages, they do participate in other research just outside of bacteriophages. So, for example, they've been doing quite a bit of work evaluating wastewater surveillance for potentially emerging infectious threats, such as SARS-CoV-2. So today, Dr. Terwilliger is going to be giving a talk that's titled The Taylor Approach, Addressing the Natural History of Infection. So please join me in welcoming Dr. Austen Terwilliger.
Thank you, David. That was great. So again, thank you for anyone here who has braved a famously rainy San Diego. Again, I'm really happy to be here, and I want to start by giving you a little bit of insight about myself. When I was 5 or 6, to show and tell, I'd bring salamanders, and then brought a dead hummingbird. I brought a parrot. I brought a lizard. So like, I've always been like fascinated with just living things and how they function and the like breadth and depth of life on Earth. And you know, I learned later the reason for that was of course Darwin's theory of evolution. And growing up in central Pennsylvania, that made some people angry, and that made me like it even more. And so yeah, I like to stir the pot a little bit when I can. And I thought, man, this is great and beautiful. I want this to be a part of my life, you know, going forward, and hopefully I can add to it in some way. So I hope you see a bit of that passion in this talk here. And we'll start with a topic that involves evolution, of course, with multi-drug resistant bacterial infections. And what we're actually fighting here, if I can get this to advance. Oh no. Might have to use the mouse to bring it back into play. There's a slide control feature here. Okay, I think it works. Yeah. So what we're actually finding here is change. De novo mutation, transduction, conjugation, transformation. These are the biological processes that subvert humanity's attempts to control these bacterial cells. And we often meet this challenge on a field that is as chaotic and dynamic in our own bodies. Right? It's our own mutations and biochemistries that just make this myriad of niche environments in which all of these processes can and do play out. So again, take some time to appreciate the dynamics and the scale that we're dealing with here, and compare that to the tools that we have historically used to address this problem in antibiotic drugs. This is penicillin. Now the chemists amongst us can play games with these parts here in blue, but there really is a limited chemical space which can be used to make new drugs. And that's why we believe at Taylor that we need to develop medicines that are capable of great change as well, which brings us to the topic and purpose of this talk. As we all know, in phage, the viruses that specifically infect and kill these cells. And they've been at this for billions of years, so they've encountered and overcome many challenges at this time, and that's why we view phage as personalized medicine for the microbe world, in line with other personalized medicines that have recently come to market. And so the challenge for our team and the field really is, you know, how to build a platform congruent with this biology we've been thinking about, right? How do you get all these teams on the same page to get a cocktail into a patient and hit what really is a biological moving target, right? Again, change is the only constant thing here. So a large portion of my talk here is going to focus on the science of how we look at phage and building a cocktail of phages, and some outcomes that we've had with patients thus far. But we'll also spend a little time on this left side as well too. All right. So what do we look for in building a cocktail? And brings us to the title of this talk, the natural history of infection. And what do I mean by this in simple terms is, well, who's there? Where are they exactly, and in what form? And how do they adapt to the ways that we try to treat and kill these cells? And we're going to drill down into each of these. So first, who's there? Not just, you know, what pathogen is infecting the patient, but also, you know, what strains are common to your group or hospital or region. We began to address that with this paper here, where we're looking for phages that are broadly lytic against any number of clinical isolates. These are E. coli from spinal cord injury patients that came to us from the VA hospital. This is just an EOP assay where the darker squares are more virulent on those strains. And we did this for E. coli, Klebsiella, and the Pseudomonas strains that are common in the VA clinic. And we also started to characterize these phages by any number of measures, plaque morphology, the virus morphology themselves, and any general info you can get from simple one-step growth curves, things like that. Again, don't pay attention to everything on here, but so we keep on growing and building our library and characterizing phage in this manner as best as we can, and these are the current phages that we have. Again, these are mostly gram-negative enteric phages with a few others scattered in there. All right, so yes, Dr. Weitzen was asking about where do we get the phages from. The overwhelming majority of them are phages. We have sans a few gram-positive phages like the Enterococcus phages you had sent. All right, so where exactly are they, these pathogens, right? Humans aren't flasks of LB media. When you get an infection, it's not uniform, right? It's in a certain tissue. It's in a niche region and environment, and we address that in a number of ways. One, building ex vivo culture systems like this urine catheter model here, where of course you have phage that are able to reduce growth in a planktonic culture, but we also have phage that are capable of getting rid of that burden again on that catheter in urine. And we're also interested in phage that work better in blood as well, and what factors might enhance their killing in blood. Again, where are they? We're interested in phages that can target themselves to where that pathogen might be. And Dr. Green, who is now with the KU Leuven group in Belgium, she had spearheaded this project where she said, "Oh, okay, so we have phage that are capable of killing E. coli in LB media, but when we use them in an animal model, or looking at killing in cecal content, suddenly our phages don't work. You aren't able to clear that pathogen from the mouse." We noticed that this was due to the insoluble portion of the cecum, and when you add NAC, which is a known chemical that breaks down mucin, suddenly our phage once more are active, and indeed you get rid of that when you add mucin back in. So it's mucin in this system that is blocking and inhibiting the phage from being able to act at that surface. So what Sabrina did was she screened our library of phage that are able to bind to mucin, and found this one ES17, which actually has a mucolytic enzyme on its tail fiber. And you can see that this phage, while not the best killer compared to our other phages in LB culture alone, it is a much greater killer in the context of this cecal environment in the presence of phage. So again, this phage is targeting itself to the region where the E. coli pathogen is found. And finally, again, looking where these phages can be, going to these niche environments to look for rare phages. Geraldo, a new graduate student in the group, pioneered this project to find the first ever lytic staph pseudintermediate phages. So just back up here a little bit. Why are we interested in staph pseudintermediates? You know, it can cause wound infections, urinary tract infections, and can infect any like open wounds from surgery as well. And it's an important multi-drug resistant zoonotic pathogen, and that zoonosis is coming from our dog friends. You know, maybe think twice when you let them lick your wounds or anything like that. So again, we were looking for lytic phages. Why? Because those are the more therapeutically viable phages right now, right? These are not temperate phages. They're not going to integrate themselves into the host genome and possibly pull back out anything that you don't want to give to the patient or that you don't want to be transferred either, like antibiotic resistance elements, toxins, virulence factors, things of that nature. But there had been no lytic phages found to date, so we were screening different canine hair, skin, and poop samples, looking for these, and well, we found some from dog hair and dog skin. They like looked clear on a plaque, so we said, okay, let's sequence these phages, and that came back, and they still looked lytic. You know, we didn't see anything in their genomes to indicate that they were temperate, and we tried to induce these phages as well in a mitomycin C assay, trying to make them turn temperate, but we didn't see any evidence of that either. So we really think that we have the first lytic staph pseudintermediate phage published, and I hope you can see at the top of the screen there on Zoom. We can't see here in the room, but DH2 on the left and DS10 on the right, and another EOP assay, again looking at virulence. Darker shaded bars are more virulent. We have two phages that cover a decent swath of the staph pseudintermediate strains we had gotten from dogs, and something that I like to think about or speculate on here again. The evolution of these phage and pathogens is like, why look at dog hair, or why source dog hair? And we think of it in terms of, you know, the lifestyle of that pathogen and what might favor a lytic phage, right? You know, I would think that a lytic phage is more favored when you have a dense amount of metabolically active cells. They know if they infect those cells in lice that they're going to find some new host. They just aren't going to leave somewhere and die. And well, you know, staph, you know, a lot of times is just kind of existing on the skin and not as metabolically active, that might favor more temperate phages. And indeed, staph strains, as a lot of us know, do have a lot of temperate phages. So we were looking at dog hair in part because, you know, staph pseudintermediate infections in dogs can start out infecting this hair follicle here, again, where you might have more dense clusters of the pathogen. So an environment like that, you know, might favor finding lytic phages. So again, we haven't done work here, but I just think it's a general cool concept, because it's general wisdom in the field that, you know, if you want to find a phage, you look to where that pathogen is. Maybe like we should focus it more on look where that pathogen is living in a way that would favor the evolution of lytic phages. So just something to chew on there. All right, in what form is this pathogen? Of course, you know, we can all find phage that just kill actively growing cells in culture, but of course, when dealing with these infections, what we're really concerned about is biofilms, right? This is EAEC, entero aggregative E. coli on a human intestinal cell line. And so we're looking for phage that, of course, are able to break up biofilm. And a former postdoc in the lab, Belkis, was responsible for most of this work. She pulled human urine, grew E. coli in urine, and then we screened our phage, added them, and seen which of them are better at being able to reduce biofilm in this setup. And we did find several of them, and more so when we moved to a urinary catheter model, again, growing these biofilms on catheters. When we mix any number of these phages, we're greatly able to reduce the biofilm and growth there. And I include this cocktail R here because it hints at where we're going next. The R stands for resistance. This cocktail was designed to cut down on the ability of the E. coli to resist phage, right? Which brings us to how do they adapt to how we are treating these infections. One, of course, is being able to overcome the resistance of phage. Keiko, a postdoc in the lab, was focused on this for a large bulk of her thesis work. Those on Zoom, I'm taking a quick sip. All right, so she challenged different strains of XPEX with one of our better phages, HP3, both in culture and in a mouse model as well, and then was looking at what came out, whether they were able to resist this phage. And well, long story short, yes. While the phage alone is of course able to kill in culture, both the isolates we got from culture and from the animals as well, from their liver and spleen, right? These were very resistant to this phage. So the challenge for us is, how can we train them to still kill these bad actors, which brings in, David had mentioned the modify a machine. We're building it with a computer user interface, so the hopes that just anyone can use it, and it's gone through a lot of different models. This is one of our older models, but just showing that it can bubble through and it can grow cells and phage. What this really is, in theory, is a modified co-culture system. So if you can imagine at time point 0, you have a host strain that is susceptible to your phage and a resistant strain that is not. Well, at first you just have your host strain that's going to be pumping out billions and trillions of phage. And again, we're just letting evolution and life take its course, right? These aren't clones. These are going to have SNPs and other changes that might allow some of these mutants to bind in favor in infecting your target strain, and if so, over time, it's going to have access to 2 hosts instead of just one host. So that will be favored through time, and you can pick out those phage. So that's that process in concept, and then actually in practice, we can get these jumps to happen in a few hours to a couple days. This is just an example of a Pseudomonas strain here, where we have a host that is susceptible and just constantly pumping out phage, as you see in this clearing on a lawn of the host, whereas this resistant strain, starting around 36 hours, you're seeing a little bit of clearing, but certainly around 60 hours, we're getting more clearing. We were able to evolve a phage to lyse that strain. So this took a couple days for us for Pseudomonas, but for E. coli and our HP3 phage, this happened in just 3 or 4 hours, really quick. And so, as you can see, this parent phage on the left here, HP3 was only virulent on a few of these strains here, whereas HP3 3.1 was able to infect all of these strains. So it became a more broadly lytic phage. And, what was really going on here, Keiko found out, and as you might have thought, it just changed its tail fiber. So we changed to a positive residue here that allowed it to bind to a more truncated version of endotoxin that's how these strains were able to resist the phage. They just cut off their toxin at the surface, and this HP 3.1 was able to bind more tightly and closer to the cell surface. Then we ran this whole setup again once more, where we challenged these resistors with HP 3.1. We got strains that were resistant to 3.1, and then we went back to the modified system and said, can we evolve, another phage that is able to infect those resistors? And we were. And what happened here was, again, just changing in the tail fibers. Whereas here, instead of binding to endotoxin, we changed the receptor for the phage, where it's actually binding OMP-C now. So again, I hope you can appreciate again like the breadth of what is possible if you're letting nature go and you bend it towards how you want things to turn out. So of course any phage or group of phages, often you're going to get resistance to this cocktail over time. But if you're able to design groups of phages in these manners that cut down on the most common pathways to resist phage, you don't see any type of resistance coming up after 24 hours. But of course, they're not just adapting to the phage; they're adapting to the drugs that we use to treat them. That brings in Carmen's work. She's just about to get her PhD, so we're very happy and proud for her. And she set up these synogram assays, which in a 96-well plate setup are really just designed to probe the landscape of the various concentrations of drug and phage, again, that they're going to encounter in your body, right? Your body is not a flask, so there's a lot of range of concentrations that are going to be going on. So again, in this 96-well setup, increasing concentration of antibiotic on the left, increasing concentration of phage on the bottom. And what we found was there's different responses to different drugs and drug classes with the same phage. So again, phage HP3, whereas the phage alone can clear out the cultures and cell, as you see on the bottom, in combination with increasing concentrations of Cipro, we're suddenly seeing more cell growth, right? So we see this as antagonism in the system. But there is hope too, right? Same phage, different drug class of antibiotic. You're seeing greater killing in the center of this figure than killing with the drug or just phage by themselves. And this is very important when moving into treatment in a patient, and something we test and screen for to ensure that at the very least the planned drug and phage treatment is not going to antagonize. If there's either no effect or some synergy rather. All right, so that's covering the natural history of infection, how we like to build cocktails. And I hope you can understand that if we're able to find and characterize phages in this manner, we can build new drugs that have these functions, right? A phage that targets itself to where the pathogen is going to be, one that's better at breaking up biofilms, one that knows in essence how the cell is going to evolve against phage and is ready for it. To probably add the synergizer on here too, right? Something that can synergize with the drug treatment. And of course, these are not necessarily going to be separate phages. Some phages can serve more than one function here, but we believe this setup is how you're able to build a standard set of phages that might be able to treat a broader range of patients. And that's what we do, and we've been working on with the Taylor system. I've gone over the right side and the bottom third of this, where how we find new phages, we can characterize new phages. Now we're going to talk about the system in practice and our work with some patients. So again, just who are we looking for here? These are chronic but stable patients that have been suffering from years, but of course modern drugs or the drugs that we have aren't working, probably because you're being infected with a multi-drug resistant bug, and that this is the sole cause or main cause of their problem. Again, trying to treat a polymicrobial infection with phage, usually doesn't work out because for how specific those phage are, or if patients have a more serious underlying health problem than the infection itself, well, you can clear out that infection with phage, but then some other the pathogen is going to just come by and take its place. But that's not necessarily bad either, right? Because whatever takes that place might be more susceptible to the drugs that we have on stock. So again, phage therapy, can be viewed in that aspect as a step towards getting patients better. There's a lot to consider, obviously, when treating a patient. So we as Taylor and the manufacturer of the phage have to be in close contact with these hospital groups, because all of this is going to say, do I have enough titer? Is it stable in the media that I'm going to be using it, or the tools that I'm going to be using? Again, does it synergize with the drugs we're planning on using? And again, change is the only constant thing. Is your cocktail still virulent against the latest pathogen you can bring from that patient? And of course, a pharmacy that can work with that cocktail for us. But when we have it in hand, we go through the typical CMC data to certify and verify these cocktails, USP testing to make sure that it's sterile and that the endotoxin content of these preps are low. Of course, we're looking at the phage genomes themselves, making sure there's nothing unwanted there. All of this and other data gets sent to the FDA for approval to treat a patient. Take another sip here. All right. So this is what I like to call the Taylor family feud. We've surveyed about 50 patients and doctors. Top five answers on the board. What are the types of infections that you're looking to treat with phage? We have joint, prosthetic infections, bacteremia, sepsis. Of course, with Simon here at IPATH, urinary tract infections and prostatitis, lung and CF patients, and any sort of device. Again, these are the most common types of infections we're being asked to help with. And again, this data is just from the first 48 asks that were sent to us. And how that broke down over time, well, some of them we didn't even get a strain for, so of course can't do a phage hunt if you don't get that strain. Ended up treating about a quarter of these patients. Some patients just got better prior to treatment. We're dealing with a lot of dire cases, so of course some people passed prior to being able to make a cocktail for them. Number of cases can get mired down for any number of reasons that I'm not going to jump into here, and some of them we just aren't able to find phage for. These are so from those first 48 asks, we were able to treat 12 patients, and what I'm going to show here is these pathogens that we targeted in these 12 cases. Again, we have most of the escape pathogens covered, and E. coli and staph several times over. We can leverage a lot of the resources that we have at the Texas Medical Center, including our TEM course. So we're taking pictures of these phages, and as we all know, pictures of phages are great. I'll just say that we have all three morphotypes of phage present here in the myovirus, cyphovirus, and the protovirus. Again, we're trying to hit a biological moving target that is constantly changing, so we're trying to reduce the time to treatment. You can see that is sort of the general trend here. And of course, this is better if we already have phage on hand that we know are safe for use, and that's what we're seeing in the gray bars here. I know it's at least a small group in the room here, but does anyone want to take a guess at what happened with case number 2 here? Why it took 2+ years to actually get phage? Much dumber. COVID. We got this strain in winter of 2019, and then all of 20 to 2021, the doctor and patient didn't necessarily want to come in for treatment, so that was why it took so long there. All right, so the outcomes for these 12 patients treated. As complicated as these infections tried to be, we tried to boil them down to about 2 main flavors. 1, were you able to get rid of the infection, and 2, did you improve clinically? Now, of course, in the second column, those field goal posts are going to move based on the type of infection you're dealing with, whether you think that that is some level of success. And as you can see down bottom, it is about a 50-50 coin flip here. Again, keep in mind what the bottom number is, right? These are patients that are chronic, have been suffering for years with no change in course. So being able to improve their infection or lives in some way, I think, is great. But let's drill a little bit down more into the types of infections that we're having some success with. Again, in this first case, you don't necessarily have to clear the infection to have an improved outcome, which we published with CIMA and IPATH here. And so again, looking at these types of infections, again, number of escape pathogens and types of infection here. So it's not just one mode. It's not like one type of pathogen and infection that we're having better outcomes with phage. It really spans the gamut. And we don't have all of the data yet in for these cases, but these trends pretty much stay the same. I'm going to focus a little bit more on this case here because that's one that we worked with CIMA and IPATH to give phage to this gentleman. Greg Breed had a prostatitis infection for several years prior, and it's great when someone's willing to tell their story. You get media crews by, and we can grab more of our team members in and just do the typical thing of looking at plaques. Again, most of the stuff we do, you can't see, so they're like, show us something you can see, show us something you can. So yeah, so of course we can bring our team members in and have some fun with that. And of course, Greg gets more quality time with his family, where, as David mentioned, coming from a basic science research background, I know that almost nothing I did there was ever going to help someone or improve someone's life. And so to move to something like this, where you see an immediate effect with their patients and family, I think is great. But of course, it doesn't end there. There's still science to do on the other side of that, and we are building that in the same viruses paper that we published with CIMA and Miley. We tested and showed as you would expect when you're injecting a high amount of protein in someone for weeks, they're going to make antibodies to that protein or phage. So serum taken from this patient at later weeks limits the infection of phage, and we're also looking at how cytokines and other analytes in the blood too will change through time, and we're hoping to build this for more patients and more of these cases. All right, so where do we go from here? This is what we call probing the cryptic genosphere, and what I mean by that is, we live in a world of phage. They're all around us. 10 to the 39th of them on planet Earth. As I mentioned, they've been doing this for 3 billion years. So, there's a lot of challenges that they solve that we would like to mine for and probe for. And so we say in lab, there's a phage for that. So we are going phage hunting. And when I started work with Taylor, someone told me, phages aren't necessarily that new or great. Any schmuck with a bucket can go to a pool and grab some phage. And I was like, I can be that schmuck with a bucket. And so, that was me with the Houston wastewater. Got some of our first phages for Taylor, but we've moved on to more scenic places too, like state parks, where we bring our team in this setup, which is pretty much a filter train that allows us to process hundreds to 1,000 plus liters of wastewater. What we're doing is that we are fractioning the water that is coming in, and then we are concentrating that fraction in which the phage and virus should be, and we can get that all down to about a 100 mil volume. So we extract the DNA from that, and we sequence what's there, and not just in 1,000 liters, but just 100 liters of these water sources. Right, we find tens of thousands of viruses, and when we look at their genomes, of course there is a lot that is known and annotated here in bottom, but there's also hundreds of thousands of genes that don't really have any known function. Right, this is what we want to probe for, to look for novel and useful functions. So we're taking DNA from these samples, making a library from them, and transforming them in a mutant background to again screen for these the novel functions. So we are doing that in a RECA background, where some of these transformants are able to rescue DNA damage back to wild type, and it's the same thought process here with our catalase mutants too. So our catalase mutant here can be rescued with some of these hits. So it's possible that we can find new and novel proteins with these functions that can help us maintain our genome, which of course has great prospects for human health. But this is just the first steps. And as David mentioned as well, what's next for us in the future? This Helix Park, which is being built, Texas Medical Center, largest medical center in the world. We always like to say everything's bigger in Texas. It is expanding even more with this park, and we're going to have some space on the third floor of this building here that is circled in red, where we're going to be building a kind of small, a 2,500 square foot GMP space that is dedicated to the purification and manufacture of phage. So of course we're going to need some cool new toys and robots for that. So that's one of the latest things that we're looking into and trying to build now. So with that, I'd like to thank all of the doctors and Sama and Miley, who have helped to bring patients to us. Anthony, as the founder of Taylor, and our friends at the FDA as well, who, to their credit, have been very open on the prospect of making phage real for us in the future. And so with that, I will take any questions. Thank you. Probably a lot on Zoom.
I'm curious, in the case where you train the phages with like the 2 bacteria and the filter.
Yeah.
Do you how often do you observe like resistance to the from the bacterial side, or do you observe like a drop in the phage titer, or like which other dynamics are going on?
Yeah. So are you trying to ask in that first chamber where the susceptible host is constantly pumping out phage, how often we see some level of mutants that are coming up there that
Yeah.
are resistant to phage? Okay. So that diagram that I had there wasn't necessarily the whole setup. There's also a reservoir of these host cells growing that feed into that chamber. So you're always getting new host cells being fed in there that oftentimes are more fit than any mutants that come up. So they're always able to keep on making phages. But certainly in that other side, in the target side, that chamber that you're looking to hit, we can certainly monitor what the optical density of what that culture is, and when that drops, yeah, that signals to us that we found some new phage.
All right. How consistently are you recording EOP values greater than 1?
How consistently are we recording EOP values greater than? Well, so we don't necessarily have a scale for greater than 1. If it's greater than 1, we put it up there and show it. But yeah, we don't have a scale past that to like say, is it 10? Is it 100? Right. It's, from my memory, it's never really that high that it's like greater than 1 to like 10. It doesn't get in the 100 like 1,000 fold range.
Yeah, I mean, it would just suggest that you're growing phage to a higher titer than on its host, which
Yep.
we do not see here very often. So
Oh,
I was
really?
wondering if like your titers are high, or like if like 10 to the 11, something like that, or are they lower than that?
So a lot of that EOP work is done with plate lysates rather than purified phage, although we can do it with purified phage. But again, purified phage is like gold. You know, you like try to save that as much as possible. So those plate lysates are like in the range of 10 to the 8th to 10 to the 10th PFU per mil. And again, if that EOP is greater, right, you're then in the range of like still like 10 to the 10th to 10 to the 11th PFU per mil.
Hi, so I had a couple of questions.
Yeah.
One is, that system, I think you call it Modify.
Yeah.
I think it's a great system. And a lot of us sort of phage researchers do something very similar, but probably not nearly as elegant as that. And I think as we look at it, the question that probably pops in our mind first is extinction. And it sounds like you're not really going to observe host extinction based on it being constantly fed bacteria, or at least on the free side, you don't see host extinction because it's completely fed. If you see host extinction on the free side, that means you just got a really good phage, probably, right? Do you observe phage extinction, or do you have something built into it so that you don't observe phage extinction?
Right. With a susceptible host constantly being fed in on the left side of that, I have not encountered losing phage. I mean, you'll certainly lose titer. Again, we haven't perfectly tweaked things so that it can just, it can perfectly put out tons of phage. But I've never seen the phage be lost on that host side. And then on the target side, if you see clearing and you found a new phage, and then you see growth again, resistance pops back up. Well then that's perfect. We're taking that and just putting that in a new chamber and running that same system again. It's like okay, you found a way to solve this problem. Now go and solve this problem again. Not keeping them mixed again because there's differences in loss in fitness. So once you find that, you take it and just put it in a new system.
That's wonderful. I have to admit, we're just like, how can we get one of those? I understand you guys patented it and built one specifically for your lab, but it sounds like something that folks would want because we're doing these sorts of coevolution experiments, but
Yeah.
people in much more crude ways. That seems like a really elegant way to do it.
Yeah. I mean, I'm not going to stand here and say, it's perfect and runs perfectly, right? There's this process of building and making it better, but that's certainly what we have in mind, that you're going to be able to run those tests better, and that you're going to be able to predict, at least in culture, the common pathways that these cells are using to resist phage and evolve and build new phage that are capable of hitting those.
One other question I had was about the staph suit intermediate
Mhm.
Work that you're doing. The way you sold it was a little bit odd to me. And probably the reason is that it's such an important pathogen in animals like dogs that, most of us kind of look at it and say, boy, this is a great veterinary test case for phages, in essence. You sold it more as hey, maybe we can use this in humans, sort of things, which we don't often think about,
Right.
to be honest with you. One of the things that was unclear to me, though, is how good were your staph suit intermediate phages against Staph aureus? It seemed like there's probably something.
Yeah, there is some. I'm not sure if I can navigate back to that. But if you're able to remember that EOP figure that I brought up, there was 3 columns. The staph suit intermedius phage was on the left side, and it only was slightly virulent, so it had an EOP of around 0.1 on only one of the Staph aureus strains tested. Everything else was staph suit. DS10, the one on the right side, was more broadly lytic against Staph aureus and Staph suit intermedius strains, and that made sense because I probably should have mentioned this, but wasn't thinking about that time, didn't necessarily have time for it. That phage, the DS10, was phage K. So of course it hits Staph aureus strains, but what was new for that, as far as I know, is that it wasn't necessarily known that phage K could make that jump to a Staph suit intermedius strain. So that seemed to be what was happening there. And of course, when we saw that, we're like, yep, just another phage K coming back up. But at least this had some cool function to it. But again, when we looked at its genome, there weren't any drastic changes. Just a few small SNPs here and there that we haven't studied and looked into further that you would think caused that change. Yeah.
So someone from Zoom, I don't know if they want to unmute themselves and ask the question. Oh, I'm going to read it for them. Okay, so their question said, did you say earlier in your talk that some of the phage mutants created in your evolution system had gained the ability to infect host cells lacking O polysaccharide, i.e. that they had gained an ability to interact with our core thanks to mutations in their tail fibers?
Thank you for that question, someone from Zoom. I think
It was M. McCone?
hopefully you are asking about the nature of the of what these LPS mutants were, and in most cases it was a total cutoff at the inner and outer core. So really, all that was left was the lipid and the polysaccharide sugars on that first chain. Everything else was was gone.
Very cool. Okay, so it was Michael McConnell from PLNU who was
okay.
asking. Say that again. Oh, Point Loma Nazarene University. Oh, we know about him. Oh,
All right.
that's great. he's doing that.
Thanks, Michael.
Yeah, great question. Well, first of all, great talk. That was so fun to get
Oh.
to see an overview. You guys have so many innovative
Thank you.
research directions. I'm really
Yeah.
glad to get to hear about it. So I have kind of like a more, bigger picture question. So like you've been showing us these examples where, in 4 hours you've got your phage turning on a dime and all of a sudden it's able to infect everything. Like how rare is that? Do you feel like, can I give you any phage and you're going to come up with a way to direct its evolution how you want? Or I mean, obviously that's not what I expect you to say, but I'm just
Right.
curious, how optimistic are you that we can use these evolution strategies and succeed often enough to have it work out?
Yeah, that system is not fully plastic. You aren't going to make cross-species jumps with a single phage, right? Most of what we're doing is just.
Sorry, I didn't expect one phage to be able to do that. In general, we're trying to get phages that will be able to affect a particular species or
Yeah.
So do you feel like the evolution is working?
Right. To do an Appleman's protocol in that manner, where you're putting a lot of phage in one tube, hoping for a lot of magic to happen, and then you can get these jumps. Yeah, we've done that on some level with wastewater for Pseudomonas phage. So for Pseudomonas that we can't find phage for, we'll put some filtrate from wastewater that we know is present. We'll put it in that system and let it run. And we find phages that can infect that strain. So what we don't know there is whether in that system you're just being able to amplify some rare phage better than you would have previously versus having some recombination effect with other phages. But I can say that we have done that. But again, to answer the question of sort of how plastic this is, right? We're doing just small strain to strain jumps. We haven't been able to do any cross species jumps really. We tried it in the off chance that hey, if this works, this is really cool. But no, we're just doing these small strain jumps. And again, the main focus for us is being able to cut down on those common pathways used.
Is it usually SNPs and tail fibers, or do you see other kinds of bigger recombination events?
Yeah, for the Pseudomonas phage, we saw some recombination events, but for the evolved E. coli phage that we've sequenced thus far, it is just tail fiber SNPs. This is why, again, I think that you could get the same mutants out at a benchtop in a lab in a small volume using common protocols. It's just that the advantage of that co-culture system is that you can pull out really rare variants if they do pop up, because you're giving them a lot of time and a lot of hosts.
Okay, there's another question from Zoom from Zimri Mena. And they asked, were all the phages in your library found in the same reservoir, or were they found in different locations?
Yes, we find these phages in many different places, and we name our phages. So we have a letter code, symbol system. We name the phages based on where we found them. So it's shorthand. It's easy for me and anyone in the group. The first 2 letters or first 2 numbers, we're like, okay, I know it's from here. So we've done, single wastewater sites in Houston. We've pooled wastewater sites in Houston, San Antonio, El Paso, and then different freshwater sources too. And for our gram positive phages, again, Those are coming from swabs of dogs and pigs and feces from pigs and chickens. So that's just off the top of my head. I'm sure there's some more, and so definitely not one source.
Thank you. There's another question from Andrew Sue. He said, what are the therapeutic doses given in your patient cases?
Yeah, so the therapeutic dose can and does differ based on what the doctor wants and the titer of cocktail that we have. I can say pretty much all of those 12 patients you saw there were mostly IV dosing of 10 to the 9th PFUs per dose that were done twice daily, morning and night.
Thank you very much. It's really a very beautiful presentation, and I think we're going to have a whole lot more to talk about in the coming
great.
weeks to months. So just thanking Dr. Terwilliger.
All right. Thanks, guys. Thanks, Zoom. If anyone can hear me on Zoom, thank you.